Hello everybody, welcome to Science Abounds, Science at Home, where we will look at scientific concepts, principles and reactions by performing experiments in the comfort of our home. We will cover chemistry, biology, microbiology, microscopy and even forensic science. Welcome to my channel. Hello and welcome to Science Abounds, Science at Home, where we're going to discover home lab magic. In today's video, I'm very excited. I'm going to be introducing you to the world of chromatography. Chromatography has become my passion. In my career, I use chromatography a lot, and chromatography encompasses a lot of different variety, comes in different forms, and chromatography is a very powerful analytical technique used today by analytical chemists, forensic scientists, clinical lab scientists like myself, and other scientists doing research, proteomics, etc. It's very powerful. So what is chromatography? Chromatography basically is a method for separating mixtures. In chromatography, a mixture is dissolved in a solvent, a mobile phase, that passes through a stationary phase. During the passage, various components of the mixture selectively adhere to the stationary phase with greater or less affinity, separating the components physically across the stationary phase. For today's video, I'm going to demonstrate the most basic, simplest form of chromatography, and that's paper chromatography. Other forms of chromatography include thin layer chromatography, liquid column chromatography, gas chromatography, and high performance liquid chromatography, and also ion exchange chromatography. Not all forms of chromatography can be done in a home lab. The paper chromatography is a technique for separating dissolved chemical substances by taking advantage of their different rates of migration across sheets of paper or chromatography paper, filter paper. It is an inexpensive but powerful analytical tool that requires very small quantities of material and can easily be done in your home lab. Other forms of chromatography that I mentioned, like gas chromatography or GC, or high performance liquid chromatography or HPLC, will require large, very expensive equipment and has to be done in the laboratory. For today's video, the purpose of the experiment is to demonstrate chromatography, and we're going to demonstrate that by separating plant pigments. Now, plants make an amazing variety of pigment molecules. As you know, they're not all green. They come in different colors: yellows, blues, purples, reds, oranges. So what's causing the colors in these plants, flowers, fruits, are different plant pigments. And we're going to look at three types of pigments in the leaves of plants, and their retention and the production of these pigments determines the colors of leaves. The main pigments that include the chlorophylls, which most of you probably have heard of chlorophyll. There are carotenoids, and there are xanthocyanins. Carotenoids are very long-chain water-repelling pigments, and they are synthesized in the plastids of plant cells. And they are responsible for producing the bright, beautiful yellow and orange colors of plants and flowers. And we're going to look at xanthophyll, which is a type of carotenoid and is a pigment found in plants and is responsible for the yellow coloration in leaves, fruits, and flowers. Unlike the other carotenoids such as beta-carotene, which produces orange or red pigments, like I said, xanthophyll give the plants their yellow golden hues. And in addition to contributing the color, xanthophylls play a very important role in photosynthesis by helping to capture light energy. We're not going to discuss photosynthesis in this video. That is not the purpose of today's experiment. But in future videos, I will be discussing photosynthesis in more depth and doing some experiments relating to photosynthesis. So please stay tuned and watch my future videos if you're interested in learning about photosynthesis. As I mentioned, another pigment are the chlorophylls, and there are two forms, chlorophyll A and chlorophyll B. These are the pigments of photosynthesis, and they are produced in the chloroplast, within the photosynthetic tissues of the leaf. And the final group, the anthocyanins, is a type of flavonoid, which is a group of plant compounds known for their antioxidant properties. And they're the pigments responsible for the red, purple, and blue colors in many fruits, vegetables, and flowers. In today's experiment, what we're going to do is we've got our stationary phase, which is our filter paper. And what we're going to do is we're going to apply an extract of plant pigments onto the paper. We're then going to place the paper into our chromatography chamber, which will contain a chromatography solvent, which then moves up the paper. And as it's moving up the paper, you will see it will start separating. You'll see the plant pigments start to separate. So let's begin our experiment. So before I get started with anything, I want to equilibrate my chromatograph chamber, my chromatography chamber, and I'm going to add 15 ml of solvent to the chamber. So I have 15 ml of solvent, and a typical chromatography solvent are things like ethyl acetate. All right, so I got 15 ml there, and I'm going to pour that nicely into my chamber. And then I'm going to put aluminum foil to seal it. Okay, and then just leave it to equilibrate while I prepare the chromatography paper. So on the chromatography paper, what you want to do is you want to mark in pencil 2 lines. You want to mark from the bottom 2 centimeters up, you want to draw a line. And then from the top, you want to draw a line that is 1 centimeter from the top. That line at the top is where you want your solvent to develop and move up to. Once the solvent reaches that line, you then take the paper out of the solvent to review your results. Don't use pen or marker to draw your lines. The reason being is that the solvent will carry the ink and contaminate your stationary phase and contaminate the mixture that you are looking at separating. So please remember to mark your paper in pencil. So I've already done that. So I've got my markings. So now what I'm going to do is I've got plant pigment already extracted. And I'm going to put it into a petri dish, a little dish. I don't need much. So if you don't have a mixture, you can buy these mixtures and buy this kit from Innovating Science by Auden, and a link can be found in my description of this video. So you can buy this already as a kit and you already have plant pigment mixture. If you don't have that, you can make your own plant mixture by simply grinding some plant leaves, green plant leaves, in a little bit of alcohol or acetone, using a pestle and mortar to get a mixture that you can liquefy enough so you can apply onto the chromatography paper. So also I have an applicator, a plastic applicator. What I'm going to do is I'm just going to dip this. I'm just going to turn this around so it's easy for me to apply. So I'm just going to dip this into the plant pigment very quickly. And then I'm going to apply it very quickly onto my paper. And I'm going to put it against the line, like so. Oh, it's not dripping. Where is it not dripping? Okay. Okay, and then I'm just going to let it dry. Okay, now I'm going to make another application. Didn't apply very well. Okay, and I'm going to let that dry as well. Okay. Waft that in the air to dry. So I'm going to put that aside for a minute or two, and then I'm going to put it into my chromatography chamber. Okay, my mixture is dried now. Had enough time to dry. So it's time to put it into the chamber. What you need to remember is you don't want to put too much solvent that it reaches your application line. You want the solvent way below your application line so that the material, the mixture doesn't dissolve in the solvent and contaminating it. So I'm going to remove the foil and then I'm going to add my paper. Okay, I'm going to put the foil back on and I'm going to let the solvent rise to the top to where I drew the line 1 centimeter from the top. So as you can see, the solvent is rising and carrying with it the mixture, and as the mixture rises up the chromatography paper, it's starting to separate out. So we'll keep this running until the solvent reaches the top line. As you can see, as the pigments and components start to separate, we're seeing the yellow and then the orange component, or the orange pigment, is starting to reveal itself as the solvent progresses up the paper. Okay, the solvent's reached the line and the chromatography is now complete. So we're going to take out the paper and just lie it down to dry. We just want it to air dry at this stage. So the quality of your separation and the neatness of your bands that you see in your separation, there are many factors that can contribute. For example, you could put or apply too much of your mixture to the application line. You could put too little. There could be imperfections in the paper, or you have too little, too much solvent. So there are many variety of factors that can contribute to the quality of your separation. So for this was just to get you started and learn the basics of chromatography. There will be future videos where I'm going to look at the separation of amino acids using thin layer chromatography and identifying the presence of different amino acids in an unknown mixture and comparing it against controls. Now, when you separate different components, you can identify specific components by the retention factor. The retention factor allows us to identify a specific component. This value compares the distance traveled by a pure sample to the distance traveled by the solvent. So it's distance by sample divided by the distance by solvent. That is your retention factor, and you can use that in identifying specific components within a mixture and comparing it against a control, or if you didn't have any controls to compare to but have known RF factor values, then by your calculations, you can identify the component. Two factors that cause pigments to move at different rates during paper chromatography is the solubility in the solvent and the varying attraction to the paper fibers, the stationary phase, which is largely determined by their polarity, meaning more polar pigments will be more attracted to the paper and move slower, whereas less polar pigments will move faster. And that is the premise of chromatography. So two keywords or three keywords you need to understand: stationary phase, which is your solid phase that your sample travels through; mobile phase, or chromatography solvent, which travels through your stationary phases and carrying your mixture and separating the different components within that mixture based on their polarity; and retention factor, which is the distance traveled by pure sample to the distance traveled by the solvent, and can be used to identify specific components within a mixture. Well, I hope you enjoyed this video and learning a little bit about chromatography. There will be plenty more videos in the future looking at different types of chromatography, and we will be doing more expanded experiments on chromatography. Please check out my Patreon page. I have all the recipes to all the experiments in my videos on my Patreon page, and I also have exclusive videos, exclusive experiments that will not be seen on YouTube, and they are made exclusive for my Patreon members. Please subscribe to my channel. It doesn't cost you anything, and it helps support me in producing my videos, keeping my channel going, and making more and more videos, which I hope you will come to enjoy. Please also hit the like and the share button, spread the word, and also please email me your comments, and please email me photos and videos of you doing your experiments in your own home. I love to exchange and see other people doing their experiments, and I would love to see us building up a community of hobby scientists. Please check out the description in my video because I list all the resources you need to do home lab experiments and all the resources that I go to for all my chemicals, equipment, and all my kits. Until next time, keep on experimenting. Goodbye, everybody.